Tool 1 enables you to identify modules of interest by testing which of the 209 modules show activity in your gene expression data. That is, it asks, are there patterns of gene co-expression in your dataset that resemble the patterns represented by the 209 modules? It takes as input your entire dataset. It's very important not to impose any cutoffs, and that's because information about genes whose changes don't pass statistical significance or are very small is leveraged here. This allows you to analyze datasets in which very few genes attain statistical significance, whether that's due to small effect sizes or small sample sizes. All you need is a list of gene fold changes for every detected gene. So let's see how we use it. Your input file should be a tab-delimited text file with two columns. The first containing gene worm-based IDs, and the second containing fold changes or log fold changes for every detected gene. Remember, don't impose any cutoffs. You can download a sample file here if you like. This is what it looks like. And then if you want, you can upload it to see some results. It should be here in your downloads folder. You just wait for that to upload. And if you're curious, the data in the sample file is a comparison of ISP1 respiratory chain mutants to wild-type animals. By default, all genes are analyzed, but we can also choose to analyze up or down regulated genes separately. For now, let's do all genes. Click Submit. Wait a few seconds for the calculations to happen, and we get some results. So on this plot, each module is plotted on the x-axis from 1 to 209. Remember, there are 209 modules. And the activity of every module in your data set is plotted on the y-axis. Modules are colored according to significance of their activity in your data set. Module activity is expressed in units of SVE, which stands for Signed Variance Explained. It measures the fraction of variance in gene expression that's explained by a given module in your experiment. For example, to take an extreme case, if a single module explained all of the gene expression changes in your experiment, you would see an SVE value of 1 or -1. Typically, though, any module that explains more than 2% of variance warrants some follow-up, and such modules are labeled on the plot. The plus or minus sign indicates the direction of module activity, hence the signed variance explained. But this direction is arbitrary. The signs are useful if you want to compare module activity between two or more experiments. If one experiment gives you a highly negative SVE for module 66, like in my case here, and another a highly positive SVE for module 66, that would indicate that these two experiments activate this module in opposite directions. So in my case here, I can see that module 66 is the most highly active one in my experiment, followed by module 47, module 169, and so forth. The information from this plot is summarized in this interactive table. Here, modules are ranked by their activity or absolute value of SVE, and this table can be downloaded here after the results are displayed. In addition to SVE and p-values, this table also displays some descriptive information about each module. It shows which perturbation from a diverse compendium of 716 different perturbations described in our paper activates this module most highly. For module 66, it is paraquat treatment, and the SVE for module 66 in that experiment is 42%. So here, for example, you can see that my uploaded experiment and paraquat treatment activate module 66 in the same direction because they have the same SVE sign. This table also shows the top 2 Go terms enriched within genes that constitute each module, separately for the 3 Go categories. Every module consists of 2 hemimodules. These are sets of genes regulated in opposite directions within a module. You can see that here on the partitioned module definition matrix in 2 different colors. So for example, for a module that represents a response to pathogen, one hemimodule may contain genes that are induced and the other genes that are repressed as part of the immune response. Sometimes the 2 hemimodules contain genes with similar functions, but sometimes not. That's why it's useful to examine genes in the 2 hemimodules separately. Note that by design, the A hemimodule is always larger. It contains more genes than the B hemimodule, so it's more likely to be informative. We also offer a consensus annotation here for some modules based on our own interpretation of the various module annotations. But we highly recommend that you examine the module annotation pages yourself because it is very unlikely that a module can be described this simplistically. You can access these module annotation pages here, Tool 3, and I will describe it in more detail in the Tool 3 tutorial. Going back to Tool 2, just to wrap up, I will just say that we can also analyze the upregulated genes separately. So remember what this looked like for all genes. So now I've selected upregulated genes only. So I would only look at the upregulated genes in my data set, and I see that it is the upregulated genes that activate module 47 and 70. And let's look at the downregulated genes. And module 66, which was the highest overall module, is activated by the downregulated genes in my experiment. So that is useful information to have.